Good morning, students. In this video, I'm talking about retroviruses, and you know that this is a interesting group of viruses, and the discovery was also very interesting. And then this is the last two virus, that is RSV, Ross sarcoma virus, and HIV in your syllabus to study about animal viruses. So, retroviruses were discovered like way back in 1981 by very few experiments, like this experiment. Ross is the name of a person, scientist, and then he was given this fowl to which was developed tumors for a long time. The tumor was there in the fowl. So what he did was to differentiate between the avian leukemia virus and the present what he discovered as RSV virus. He took that infected tissue and then applied it to the other tissues and also grown that in cell lines. So he found out that it was like within 2 weeks there was appearance of new tumors, and then the cells were also turning into cancer cells. There was transformation of the cells, and which was not seen in case of avian lymphoma virus, which took several months to develop tumors, as well as the cell culture also. It was very late to develop the tumors. So that is how he discovered what is called as the virus was named against Ross, that is Ross sarcoma virus, and for which he got a Nobel Prize also. RSV is known for many such discoveries which made a breakthrough in molecular biology. The center dogma, what we say, the DNA to RNA to protein, was questioned by these retroviruses group. So the next Nobel Prize was to people, Temin and then Baltimore. Temin was the person in 1964 only. He proposed a DNA provirus hypothesis, telling that the RNA, using the inhibitors of RNA synthesis and DNA synthesis, that telling that there is a DNA intermediate which is called which he called it as DNA provirus, and then the viral is completed with the RNA genome. It was not accepted in those days because they questioned the central dogma. But until the famous scientist David Baltimore, he isolated the reverse transcriptase enzyme and proved that there is a DNA intermediate, for which both of them got a Nobel Prize in 1975. One more Ras sarcoma virus giving a Nobel Prize is the discovery of the oncogene, the viral part of the oncogene, and the cytosolic oncogene, that is called as the SRC gene, which yielded a Nobel Prize to two scientists, Bishop and Varmus. And the last slide of this presentation on Ras sarcoma virus also has many discoveries which were done on this Ras sarcoma virus, which led to understanding of the retroviruses. So I did not mention one point, that these retroviruses are divided into two classes, which are called as simple retrovirus and complex retrovirus, based on the mode of infection and also how many proteins are involved in this retrovirus, the whole productive cycle. Ross sarcoma virus comes under the simple retrovirus, and HIV, human immunodeficiency virus, comes under complex retroviruses. Ras sarcoma virus belongs to alpha retrovirus genus, and HIV belongs to lentivirus genus. So coming to the virion structure, as I told, it is a simple retrovirus, and it has only three proteins which are very important: the GAG protein, the Pol protein, and the envelope protein. There are in complex retroviruses, apart from these three proteins, you have another few proteins which are called as accessory proteins, which are very important for their function. So it's an envelope. The Ross sarcoma virus is an enveloped virus. You have the lipid bilayer, which is nothing but the cell which gets infected by RSV. This lipid bilayer is taken from that cell, and this membrane, the lipid bilayer membrane has some proteins which are there. That is one complex protein is the transmembrane protein attached to the surface protein, which is important for the infectivity. Below that lipid bilayer, there is a lining of matrix protein, inside to which you have the capsid or the protein coat of the raw sarcoma virus, which includes two RNA particles which are coated by nucleocapsid protein. And then in that freely there are the other three protein which are called as PR, which is the protease, and then you have the integrase ion and the reverse transcriptase protein are packed inside this capsid. The RNA genome is in two copies, and then the two copies contains the tRNA which is attached to the RNA. So this forms the infective particle of sarcoma virus. So coming to the genome structure, the genome of viral RNA is single stranded RNA genome, and then which is coated with nucleocapsid protein, which is about 8 to 10 kilobase space in length. And there are only three ORFs, that is GAG, coding for GAG protein, Pol protein, envelope protein. These are the main three proteins, and there are cis acting elements at the 5' non-coding region, and that is the R region, and the U5 and PBS stand for primer binding site. And there are three cis acting elements in the 3' non-coding region, that is PPT, polyuridine tract, polypurine tract, sorry, and U3 and R region. R region is very same at the 3' end and the 5' end. It's important during replication. So you remember this. And there is 5' and 3' splice sites which are important for the splicing. And then you have the packaging signal also, which is also very important, which can bind to the RNA and help in packaging of the which binds to the other protein, that is the GAG protein. And during the binding and assembly, this sequence is very important. Another important feature is the 5' end of this RNA is methylated. The guanosine is methylated, and 3' end is polyadenylated. So it can act directly as the mRNA in a eukaryotic cell. So how does this binding and entry happens? The life cycle begins with the recognition of the receptor on the probable cell, and then the receptor is a glycoprotein, which is simplest glycoprotein of 72 amino acid, which is can be a glycerol, glycosyl phosphatidyl inositol linked glycoprotein, which has a domain which is similar to LDL receptor binding repeat, the extracellular domain of this receptor protein. And that is very important for this receptor is called as TVA receptor on the cells. Okay, the surface SU protein which has the transmembrane domain makes an interaction with this TVA protein on the host cell, and once this interaction happens, the next step is the inhibitory peptide and the fusion proteins are released from this subunit protein. So okay, the surface protein making an interaction with the cell receptor, and the cell receptor is a TVA protein which makes a conformational change, and then the fusion peptide is released, which makes an binding to the membrane, and then you have this formation of hinge like region, that is the fusion happening. Once this membrane fusion happens, because of the low pH and low temperature, there is formation of the restricted structures which are like at the physiological pH can open up because of the pulling of this inhibitory peptide complexes. There is opening of the membrane after fusion, and the viral capsid is released into the cytoplasm. Once the fusion happens, the viral core enters the cytoplasm. Reverse transcription is initiated within the viral cores. Okay. The result is double-stranded DNA copy transported to the nucleus and integrated into the host chromosome. Now, before going to integration, this is a complex process where the it is carried out by reverse transcriptase. You remember there is a in the structure there was two tRNAs attached to two RNA molecule. They are very important because they act as a primer. The tryptophan tRNA, which is annealed, gets annealed to the primer binding site near the 5' end of the genome, the first step. And to that complex is the reverse transcriptase, binds to viral RNA and begins the synthesis of DNA using RNA as a template. DNA synthesis continues till the 5' end. Now 5' to 3' is the synthesis. So once the primer binding site tRNA, which is acting as a primer, continues till the edge, that is where it stops. And now it's an important step because the RNase H component of the reverse transcriptase, RNase H, which is the part of the reverse transcriptase, degrades the RNA strand of the RNA DNA hybrid located upstream of the primer binding site. Now, after this degradation, you have the small synthesized DNA and you have the remaining RNA. Both are attached at the primer binding site. Now, the first shift happens. That is the first strand shift. This, which is bound RNA DNA complex at the primer binding site, is called as minor strand strong stop DNA. Okay, now the shift happens. Now when I was telling about the genome structure, I have told that there is R sequence at the 5' and R sequence at the 3' ends. Okay, now the first strand transfer occurs next when the minus strand strong stop DNA jumps to the opposite end of the RNA and hybridizes to the complementary repeated sequence R. RNA dependent DNA synthesis proceeds from the R through a region known as polypurine tract, then continues to the primer binding site, which is at the end of the RNA strand. The polypurine tract is not digested. You can see here, after synthesis, polypurine tract is not digested effectively by RNase H and remain as the final vestige of viral RNA template. The RNA PPT sequence forms a duplex. Now, after the shift and the synthesis, you have the degradation of this RNA at the 5' end, but the PPT sequence still it is attached to the DNA. DNA synthesis using the nascent minor strand of DNA as a template continues. That is, DNA dependent DNA synthesis continues till the end of the minor strand. A segment of annealed tRNA primer. Till that, the DNA synthesis will continue, terminating at PBS sequence and forms the first positive strand, that is it forms the plus strand strong stop DNA. And then the primer tRNA is degraded by RNase H, leaving the primer binding site intact. Now you have this extension which does not have the primer binding site, which was the tRNA. And now this green color, which is newly synthesized DNA, which has the primer binding site, which is like a single strand overhang. Next is the second strand transfer, involves translocation of the minus strand primer binding site to the opposite end of the DNA duplex. Annealing of the complementary positive strand primer binding site happens, and directed DNA synthesis continues until both the strands of the provirus are fully intact. In RSV infected cells, synthesis of the double strand DNA provirus is not finished until the reverse transcription complex enters the nucleus. The completed product of reverse transcriptase may be integrated into the host genome, or the blunt ends forming the lateral terminal repeats ends may be joined by the host ligase to either form a 2 LTR circle or a single LTR circle by auto-integration or by ligase, or it can be integrated into the host genome by integrase enzyme. Now, integration preferentially integrates the viral DNA in regions of the genome that encodes gene transcribed by RNA polymerase II. Now, that is important because the promoters helps in easy transcription of viral DNA. Following transcription of proviral DNA, which results in the synthesis of genomic length viral RNA, which is processed to the cellular mRNA to contain, okay. So this proviral DNA, which is integrated into the viral genome with the help of RNA polymerase II, it can synthesize its required mRNA. It can be full length unspliced RNA which contains the GAG pol both the sequence. Also it might be spliced to form a specific protein like envelope associated protein or SRC. It's very important that this protein without the processing has to be exported from the nucleus through like other spliced mRNA, how they are exported to the cytoplasm, like that these also has to be exported to the cytoplasm. The unspliced viral mRNA has two potential. They can act as a template for translation of structural protein, that is the GAG and GAG-POL protein, and the other is it can act as a genome for encapsulation into assembling virion. Now this pre-mRNA are not exported to the cytoplasm to make GAG and GAG-POL protein simply, but they are exported by the machinery which is used by cellular mRNA, that is TAP and DBP5 complex, which helps in the transport of viral unspliced mRNA by binding to cis acting direct repeats. Now I'll repeat once again. This viral genome which is integrated into the nucleus, which is close to the RNA pol II promoters, gets transcribed, and during this transcription it can be spliced directly into envelope associated protein coding RNA or SRC coding RNA, or the GAG and GAG-POL are usually produced as unspliced mRNA, which the mRNAs which goes for translation and produces GAG and GAG-POL are basically synthesized as unspliced RNA. So this unspliced RNA has to be transported to the cytoplasm. The normal function of the eukaryotic cell is unspliced mRNAs are not sent to the cytoplasm. But if this direct repeats which are present in this mRNA can interact with the cellular complexes such as TAP DBP5 complex, they can be sent into the cytoplasm easily. So in the cytoplasm you have synthesis of this GAG protein and other proteins synthesized. Now this GAG protein is very important. They can be broken down into matrix protein, capsid protein, and the nucleocapsid. Pol protein has the core reverse transcriptase activity, and it has the integrase, and there is also aspartase protease um, part. Envelope protein contains two regions, that is the surface protein, that is GBP 120, which is help in receptor binding, and transmembrane protein, that is GB glycoprotein 41, which is the fusion domain. Now this GAG protein is very important because this GAG protein has some important regions. This GAG protein is synthesized as polyprotein. It is not the final GAG protein what is in the cytoplasm. The one which gets integrated into the provirus is the GAG polyprotein, which is cleaved during viral maturation to generate MA, that is matrix protein, P2A, P2B, P10 protein, and then Ca, that is capsid protein, Sp, a small one, that is spacer protein, and Nc, that is nucleocapsid protein, and Pr is a protease protein. So the GAG contains also NLS, that is nuclear localization signal, at the N-terminal end and at the C-terminal end. The NLS in the N-terminal region of that is present in the matrix protein region binds to some important host factors such as importin 11 and transportin or SR or transportin 3. The NLS in the NC, that is nucleocapsid region, interacts with importin alpha adapter. You remember, this is very important for getting into the nucleus, importin alpha beta complex. CRM is very important to get exported from the nucleus. So the proteins, once they are synthesized, they can be imported also, some of the GAG protein. Now the NLS in NC interacts with importin alpha adapter, which in turn bind to the importin beta to create functional nuclear import complex. The NC region, that is nucleocapsid region, also contains a zinc knuckle domain composed of cysteine histidine boxes. This is responsible for binding to genomic viral RNA for encapsulation. There is also a CRM export factor binds to NES, that is nuclear export signal in P10 region of GAG. So the major homology region of GAG is important for its structural role. So there is the structural proteins are MA protein, matrix protein, capsid protein, spacer, nucleocapsid. The membrane binding domain M in the GAG protein folds into an as an alpha helical domain that transports the GAG to the plasma membrane, and I, you can see this last NC where it is I, or the interaction domain of NC is present in 2 copies and is made up of cysteine histidine boxes, and the cysteine histidine boxes, that is like the zinc knuckle domains, and basic residues. The zinc knuckle, that is cysteine histidine boxes, are in black, and then the red, that is the basic residues, are in red color. Those are the what they are interacting with the viral RNA as well as nucleocapsid interaction. The late domain, the L domain, that is near the P2A, P2B, and the P10, basically P2B. that is the P2B region. The rate domain is in the P2B region and interacts with a complex of host factor including ubiquitin ligase, NED4, and several members of ESCRT2 and 3 families that facilitate the final pinching of step of the budding process. So this GAG protein is a very important protein showing so much of interaction. So now the part of the protein can interact, that is this NC domain can interact with viral RNA, the other proteins can interact with the membrane matrix protein. So finally they are like assembled as the provirus. So now coming to this picture, now you can see this is the provirus or immature virus, which has a central core of viral RNA, and then the pink one are the GAG-Pol precursors, and the zigzag black line is GAG precursor, which will be later broken down by this protease. You have the lipid bilayer, which has nothing but the host cell pinch off, and then you have the envelope protein, which also will be like in the matured form very clearly into two subunits. The envelope here is derived from the host cell plasma membrane when the virus particle buds off from the host cell. Envelope glycoprotein, the surface protein and the TM components protrudes as spikes from the virion envelope and attach to specific receptor on the target. Viral particles are assembled within the cell as immature particles which are not infectious. The morphology is very similar, not very different, but internal the GAG and GAG-Pol layer is what gets changed in the final mature particle. With budding, the immature particle undergo maturation, a process catalyzed by retroviral protease enzyme to generate the infectious virus particle. Now this protease is present as fusion with GAG polyprotein, which is unique to RSV. During processing, the viral protease dimerize and cleaves the immature GAG polyprotein into matrix protein, capsid protein, nucleocapsid proteins, as well as P2, P10 and spacer peptides. So this forms the final mature particle of the RSV virion, and that is ready for infection to the other cell. So to overall cycle of the RSV is infection is initiated with binding of the viral glycoprotein to the cellular receptor, triggering fusion of the virus and cellular membrane. The viral core enters the cytoplasm and reverse transcription occurs within the pre-integration complex in the cytoplasm. The pre-integration complex is translocated to the nucleus and nuclear entry occurs with nuclear envelope breakdown during mitosis. Also there are reports that without breaking down the nuclear membrane in mitotic cells also the viral particle can enter into the nucleus, and also through nuclear pore in non-dividing cells. Integration into the chromosome is mediated by integrase enzyme and facilitated by other host factors. Like ligase is needed. Such kind of host factors are like helping in integration into the host genome. The proviral DNA is transcribed by RNA pol 3 and genome length viral RNA are made. A subset of these mRNAs undergo splicing and exit the nucleus for transition into envelope and SRC protein. Envelope protein is synthesized in endoplasmic reticulum, transported via vesicle to the plasma membrane, and it resides in the plasma membrane. And there are SRC protein which induces the to an oncogenic state, that is the mitotic state, so that more and more of these particles are uh, produced. There are unspliced viral RNAs also, which are sent, that is the GAG and GAG-POL, which all exits from the nucleus, and in the cytoplasm you have the synthesis of this proteins, and these poly proteins are then taken over to the membrane, where the GAG multimerization happens, and then this multimerization also takes the viral nucleic acid, because there is a viral genome RNA genome binding domain in this GAG polyprotein, and then this budding happens. Budding is like immature particles, and it produces the maturation or mature particles that are ready to infect the another cell. So this is the whole productive cycle of Ross sarcoma virus. So Ross sarcoma virus has been known for many of the discoveries, like in 1910 a spindle cell tumor, that is where it started finding out of this Ross sarcoma virus, and then followed by tumor transition by filtrate. Okay, tumor was transmitted by filtrable virus that fulfill Koch postulates, and also development of a method to measure the titer of infectious RSV was also many such new invention and discovery was done by using this Ross sarcoma virus. So the next virus is the complex retrovirus, which belongs to the genus lentivirus, which is human immunodeficiency virus, as popularly we call as HIV. So it's a very important retrovirus that belongs to the complex retrovirus family, because it codes more than the 3 important genes, other accessory proteins which are very much required for viral propagation and pathogenesis. So HIV epidemic was in during the 1980s, where like currently more than 37 million people are infected with HIV, and during the 1980s the important western countries, which are United States and Europe, was completely infected with this virus. And okay, the first reported case was in 1981 among homosexual living in Los Angeles, and the epidemiological characteristics of this disease drew a lot of attention, because they were affecting the immune cells. The disease was named as AIDS, which is acquired immunodeficiency syndrome, reflecting the immune deficiency among the individuals. Like 2 years later, these two important scientists from their French virologists, Lou Montanger and then Francois Bercinois. So they were the people who first gave the first clinical report on what is this virus, and how this is infecting. So we all know that HIV infects the cells which has the receptor CD4 plus. CD4 plus is a characteristic of T lymphocyte, that is helper T cells, which are present in peripheral blood. So these T lymphocytes in immunology, later you will study that they are very important key players in presenting or in the immune protection pathways. So because there is depletion of this cells, there is immunodeficiency. So as a result of this immunodeficiency, there can be secondary infections also, which adds to the severity of the viral infection. This belongs to the again I am repeating, this belongs to the class of complex retroviruses of the genus lentivirus. Besides HLV, there are other viruses also in this group called as HTLV1, which is the first human retrovirus which was discovered in lentivirus group. And there are simian immunodeficiency virus. It's interesting to know that simian immunodeficiency virus cannot infect the humans, and humans cannot infect the monkeys. Coming to the virion structure, so this is the structure. Again, it's an envelope protein. It's looks very similar to that simple retroviruses, but the characteristic feature is how the capsid is like. It's like a cone shaped capsid, and which is enveloped, and the envelope contains the spike like elements, which are nothing but GP120 and GP41 heterodimers. And inside this cone shaped capsid, you have two copies of the genome. You have a copy of a genome, that is the RNA, and then with that you have the reverse transcriptase and integrase which is attached. There is protease also which is included in this virion particle. Apart from this, you have accessory proteins which are there, which are like VPR, and we'll see other few accessory proteins which are very important for the infectivity. Coming to the genome, HIV is grouped into two genotypes, HIV1 and HIV2, based on the some critical proteins which are absent in, though the function is same, there one of the protein which is present in one and it is not have to absent in two. So the genome of the viral RNA is around 9 kb in size, which encodes for the main protein, the gag protein, the pol protein, and the envelope associated protein. It also encodes other 6 accessories protein, that is TAT, REV, NEF, VIF, VPR, and VPU. The genome, when they are like encoding this protein, when transcription happens, they are spliced into two classes of subgenomic RNAs, that is 4 kb singly spliced RNA and 2 kb doubly spliced RNA. That's very important because this doubly spliced RNA is what is the Rev protein, and which binds to the RNA and other singly spliced RNA, and which helps in the mobility of those RNA. Later in translation, I'm going to talk about this. The singly spliced RNA encodes for envelope, VPR, VIF, and then VPU proteins. Doubly spliced RNA encodes for TAT, REV, and NEF protein. All 6 accessories proteins are encoded by subgenomic RNA. Now, HIV-1 is one which is responsible for current global HIV epidemic. HIV-2 causes AIDS in regions of West Africa. Overall, the genome structure of HIV-2 is almost similar to HIV-1, except the VPU protein is not found in HIV-2. Instead, VPX protein is found in HIV. So here what we are focusing is on HIV-1. So coming to the life cycle of HIV, again there is an entry, there is transcription and translation, and then you have packaging. So it's something very similar to the simple retrovirus, Ross-Sarcoma virus, what we studied earlier. There is receptor mediated endocytosis in case of the simple retroviruses, but in case of HIV, it is via direct fusion. So in case of Ross sarcoma virus, there was receptor mediated endocytosis, and that fusion protein, what I was telling, the surface protein which binds to this TVA receptor and then actually pulls apart the receptor because of the low pH and low temperature, is the drivers for low pH is the driver, low temperature adds on, which pulls apart the lipid membrane, and then you have the release of virion. But here it is very simple. It interacts with the CD4 receptor of the helper T cells. Along with that, CCR5 is the coactive, like it's a chemokine receptor. It's like co-receptor interaction induces, I mean, there is direct fusion and release of the capsid inside the cell. Now it's very important, this chemokine receptor, because once the viral particle is released inside the cell, there is actin network there which prevents, or it acts as a barrier for movement of this capsid. But if there is interaction with the CCR5 co-receptor or the CXCR4 co-receptor, either in the T lymphocytes or in the macrophage cells, now there is actin depolymerization happen. The result of this actin depolymerization is the actin dynamics is disturbed, and then this capsid can easily go inside the cytoplasm by cytoplasmic trafficking. The CCR5 is in macrophage, CXCR4 co-receptor is in CD4 plus helper T lymphocytes. So once it is entered into the cytoplasm, this is entry is basically non-dividing cells. You have the complex formed where you have the synthesis like the Ross sarcoma virus. The reverse transcriptase will be activated once the capsid is released, and you have the genomic DNA present in the cytoplasm. Then it can enter as well as, like, there is reverse transcription and they enter the nucleus. But HIV has the ability to get into the nuclear pore of non-dividing cells. There the dividing cells were very important for Ross sarcoma virus, but here HIV can directly enter the non-dividing cells. So that is like they are in the resting phase. And reverse transcription, I will not repeat again. It is the same mechanism mediated by the tRNA, and then you have the first strand small stretch tRNA. The tRNA binds at the primer binding site, and then you have the first short DNA synthesis, and then RNase H activity cleaves down the DNA, and then you have the shift of this tRNA to the edge of the R site, which are same at both the ends, and then you have the synthesis of first strand DNA, and you have next is RNase H activity, which breaks down the RNA in the RNA DNA complex, but till the only the polypurine tract PPT region, and then that stretch of RNA initiates the DNA synthesis till the PBS site, till the tRNA which is recognized in the PB site and binding. Then the tRNA is removed by RNase H digestion activity of the retrovirus. Then you have circularization, and then it can circularize and shift also. Also this strand is taken to the other end, and you have the complete synthesis. This replication or making this multiple copy of R and PBS is the lateral terminal repeats, and using the integrase enzyme, the DNA, double stranded DNA is integrated into the genome of the virus, of the infected cell. Now comes to the transcription. The viral pro DNA is integrated in such a place that there is easy transcription of the viral gene, and the viral genes In addition to GAG-POL NV polyproteins, it also encodes six important proteins which are helping in the pathogenicity of the virus, which range from 10 to 25 kilodaltons. And in terms of regulation and viral pathogenesis, these extra proteins are very important. So the first important protein what we'll talk about is the TAT protein. So this TAT protein stands for trans activator of transcription, and it is an important transcriptional trans activator essential for establishment of HIV infection. So why it is important? Because it binds to an important RNA region, that is the RNA binding. It's a RNA binding protein which specifically recognizes a RNA stem loop structure in HIV termed as TAR, that is the TAT responsive elements. Here you can see a secondary structure, that is the TAR, that is TAT responsive element. Trans activator of transcription binds to this TAT sequence and then to other important transcriptional factors. The tags bind to RNA. It's very important because if the TAT is not present, RNA polymerase falls off the template immediately after transcribing TAR sequence. So if this TAT is binds to the TAR sequence, which brings down two important factors, that is CDK9 and cyclin CDK9 and cyclin T1, which forms a complex called PTEFB complex, that is positive transcription elongation factor, because this improves the efficiency of RNA polymerase 2, allowing transcription of full length viral RNA. So overall this TAT acts as a anti-terminator of viral RNA transcription. Okay. So without TAT there is abortive transcription, but with TAT you have full length RNA transcription. So coming to the next protein, that is the REV protein. So here you can see the doubly spliced 2 kb RNA is what is representing the REV protein. Once it is synthesized, this in the cytoplasm, it's translated, they come back to the nucleus and they regulate important function. The pre-mRNAs of eukaryotic cells are exported out of the cytoplasm following the excision of introns through RNA splicing. The complete removal of introns is very important for the nuclear export of pre-mRNAs. In case of retroviruses, not all subgenomic RNAs, also the genomic RNA contains the introns. They are not spliced. So they need a mechanism to bypass this and then go into the cytoplasm. That is what is regulated by this REV protein. REV stands for regulator of expression of virion protein. And during evolution only this protein have been integrated into the virus. REV protein is an important RNA binding protein that binds to an RNA sequence called as RRE sequence. Like the TRE sequence, this is also called RRE sequence, meaning REV responsive element. So you can see at the 3' end this blue color protein is bound, and that is the REV protein at the REV responsive element. So it's an important small protein which can cross over the nuclear membrane via the nuclear pore in an CRM1 dependent manner. The Rev protein binds to the RRE elements which lies in middle of the second intron and carries the intron containing RNA to the cytoplasm via protein export mechanism. This unspliced RNA are transported to the cytoplasm. Okay, this is the function of Rev protein. Now coming to the next protein, you have three next protein which is part of this is the ubiquitin E3 ligase, that is VIF, VPU and VPR. VIF stands for virus infectivity factor. Now this is a very important protein because it can phosphorylate it can ubiquitinate some critical factors like the first VIF will take. It can ubiquitinate the host restriction factor, which is ApoBec3G. So what is the function of this protein? In the presence of VIF factor, that if a wild type, you can have two type of virion particle without this VIF gene and with the VIF gene. First I'll tell about with the VIF gene. If the presence of VIF gene, the restriction factor which I spoke last was ApoBec3G gets degraded. Hence, there is no package of this restriction factor into the virion particle. When this packed virion particle infects the other cell, it is a positive infection. If you have a virion particle which is defective of this VIF gene, then when the virion particles are packaged with other structural protein, this restriction factor also get packaged into the HIV particle. When such packaged HIV particle infects the other cell which contains this host restriction factor, it causes when the release of genomic RNA is done, there is deamination of cytidine to uracil during the entry that results in hypermutation and degradation of the viral DNA. So hence viral infection is blocked. So important is VIF gene for the virus because that leads to ubiquitination of this important restriction factor and then breaking down. So next, coming to NEF gene which was named as negative factor. It is NIF is a myristoylated protein associated with cytoplasmic membrane. So it is an important protein because it helps in maintenance of viral load and accelerate disease progression in HIV infected individual. So it downregulates multiple plasma membrane protein including the CD4 receptor, MHC class 1 receptor and CXCR4 receptor. Why? If the virus are releasing from the same cell, they either should not be bound to the same cell and getting inside, or if there is cytoplasmic production of this MHC and CXCR4 receptors, the virus tend to bind that. So the receptor downregulation has an important immune function. This prevents the virus infected T cell from cytotoxic T cell lysis via downregulating the MHC class 1 molecule. MHC class 1 molecule is the one which produces the viral peptide to the cytotoxic T cells, that is CD8 plus cells, and leads to lysis, that is impaired. Second is impairing helper T cell function via downregulation of CD4 plus and then CXCR4 receptors on the membrane. in addition to the CD4 plus down regulation, the main which is the main receptor for HIV, enables enables the HIV to avoid GP120 CD4 interaction, and if it is interaction, it is retained inside the cell without exiting out. So that is viral release also is prevented if the CD4 receptor is not functioning. So the important factor of this NEF protein is down regulation of the CD4, MHC class 1 and other co-receptors in the cell. So coming to next protein is VPU protein. Here the center one, which is also a part of a E3 ligase. VPU stands for viral protein unique. It is an integral membrane protein, and it has it interacts with important host factors, and which affects the viral assembly and release. The first interaction of the newly synthesized CD4 receptor in the endoplasmic reticulum is VPU serves as an adapter for recruiting this CD4 plus for degradation. That means VPU is required for efficient viral particle release because there is one important plasma membrane protein, that is tetherin, which blocks the viral release. So what this VPU does is it ubiquitinates the CD4 receptor as well as tetherin. So the like interaction of further GP120 CD4 complex is not formed, and then you have this viral release block protein tetherin, which is also ubiquitinated by this E3 ligase, is broken down by proteasome. The tetherin protein after ubiquitination is broken down by proteasome. The next protein is VPR protein. It is virion associated factor. VPR stands for virion protein R. VPR is found packaged inside the capsid. Around say 100 to 700 copies can be there inside the capsid. VPR is not required for HIV propagation in dividing cell, but it is required in non-dividing cells such as macrophages. There are multiple activities associated with VPR, that is one of which is arresting the cell at G2M phase or induction of apoptosis. Okay, the important factors which are associated with such functions like induction of apoptosis and G2M phase arrest is done by the VPR. It is also a Cullen based E3 ligase. It interacts via DCF1, and then it ubiquitinates many protein like this UNG, uracil DNA glycosylase, which is importantly involved in DNA repair mechanism. Also proteins of ATR, that is it's an ATM related gene. In the other viruses I have mentioned this double strand DNA damage recognition system. In that ATR is a serine threonine kinase that is important in sensing DNA damage and activating DNA damage checkpoints, leading to cell cycle arrest. So that such factor also are ubiquitinated, and the cell cycles are regulated. So this is about the regulation of transcription. And then you have the once the regulation is happening, you have all the transcription of the required proteins, that is the GAG proteins, and then other important VPR, VPX, VPU, all are synthesized by this host factors. Host cytoplasmic translation by the ribosomes, and the viral RNA is also made by full length RNA using the rev protein. They are transported or exported to the cytoplasm. Now the packaging happens, the GAC envelope protein, associated protein, are synthesized in the endoplasmic reticulum, and then they are sent to the membrane, where they are assembled at one particular loci. Once they are assembled, the other proteins, the capsid proteins and the viral RNA polymerase, and you have other important accessory proteins which are packaged into the virion, and the release of the virion is through budding. So there are many host restriction factors. Once again I am summarizing: host restriction factor that are targets for the virus, because they help in maintaining the cell, like protecting the cell. So important was the WIF factor, which interacts with the APO BEC3, and they help in not involving this into the packaging. VPU, that prevents the tetherin from holding on to the virus at the plasma membrane. This tetherin is one of the interferon stimulated genes that prevents the release of virions. So the VPU has an important function helping in release. There is also two important additional host restriction factors which were also discovered during the study of this HIV, that is TRIM5 alpha and SAMHD1. So TRIM5 alpha 21 or alpha is TRM5 alpha is a very important host restriction factor responsible for determining the host range. Okay. So there is no cross reactivity because of this TRM5 alpha. SMHHD1 is also important because it shows nuclease activity, specifically the ribonuclease activity, and this VPX again is relied on SMHHD1, which mediates the block to release reverse transcription by down regulation of this SMHD1. This VPX E3 ligase also phosphorylates this and breaks down. And another important, these were the main important factors, that is tetherin, SAMHD1, and then APOBEC3, which are like confers the intrinsic immunity of the host cell against HIV. To summarize, retroviruses are into two groups: simple retrovirus and complex retrovirus. The complex retrovirus has many more proteins than the simpler retrovirus. The virion structure is enveloped, and there is dimeric RNA genome. Three important viral proteins are there, that is reverse transcriptase, integrase and protease, which are packed inside the capsid. The viral genome is that is 8 to 10 kilobase pair RNA having a cap 5 prime end and poly-A tail at the 3 prime end. It encodes for important 3 GAG, POL and NV proteins in case of simple retroviruses, and many other splice variants of these three important RNAs. Genome replication happens with the help of reverse transcriptase. In case of lentivirus, which the HIV is the prototype, contains 6 accessory proteins, and then host restriction factors like TRIM 5 alpha, APOB, C3, tetherine, and SAMHD1, which were discovered during this study of HIV. Excessive proteins of HIV counteract these restriction factor to establish the HIV infection. So this finishes the animal viruses part.